The overall goal of this procedure is to prepare an orthotopic mouse model of pancreatic cancer that uses in vivo bioluminescence imaging to non-invasively monitor tumor development. This is accomplished by first resuspending luciferase-tagged cancer cells in Matrigel. The second step is to perform a laparotomy to open the abdominal cavity and to locate and secure the pancreas. Next, a suspension of tumor cells in Matrigel is injected into the pancreas. The final step is to replace the pancreas and use sutures to close the abdominal cavity. Ultimately, bioluminescence imaging is used to monitor tumor progression and track response to therapeutic interventions.
The main advantage of this model over in vitro-based cell assays or subcutaneous models of pancreatic cancer is that it really enables us to investigate the interactions between the pancreatic tumor cells and the pancreatic microenvironment. The kinetics of disease progression in this model are highly reproducible and occur in a short time frame, making this model useful for the examination of novel therapeutics in pancreatic cancer.
Visual demonstration of this model is critical, as injection of tumor cells has to be performed carefully to prevent leakage.
To begin this procedure, culture pancreatic cancer cells that have been transduced to express luciferase, as is described in the written protocol, until they are